This recorded session is dedicated to our lecture on the animal excretory system. This will be part one of our last module for the final term on the urogenital system. So, we're covering for this session the excretory system of the animal body. So we'll talk about um, the general structure of the excretory system in both vertebrate and invertebrate animals, and we'll give emphasis on the excretory system of humans. So, um, in the previous modules, we've covered key organ systems of the animal body, namely the muscular system, the digestive system, and the respiratory system. And you would remember that the different parts of those systems across all animals, of course, will have specific functions that are dependent on a stable or dependent on the stability of the animal, the stability of the environment, or essentially the stability of the animal body where the organs are housed in. And looking into cells, living cells of an animal, unicellular or multicellular, will require a stable medium in order for it to perform all of its functions, from growth to metabolism to reproduction. So, all of those essential physiological activities will require the maintenance of a stable environment. No? And um, for the case of animals which are um, heterotrophic by nature, we expect that excess material, excess nutrients that are not used during or that are not used entirely by the animal, those extra nutrients will be converted to waste material that are not needed, and if are not expelled out of the animal body, will threaten or will jeopardize the stability of the internal environment of the animal. And the excretory system carries out the special function of filtering material that are not needed by the animal. So you can simply look at it as an organ system that is responsible for cleaning up the animal body in order to ensure that homeostasis is maintained. And some of the key things that are associated with a stable environment, a stable internal environment of the animal, include the maintenance of correct or standard cell concentration of fluids, the maintenance of a normal pH, especially for the blood, the pH of the blood and pH of internal tissues. In our discussion on the respiratory system, I think I'm sure I've mentioned or discussed about how the body regulates respiration chemically, or by means of chemical signals, and primarily the blood's pH is a driver, primary stimulus for the animal to regulate respiratory rates. Also, we relate the removal of metabolic wastes with the maintenance of an internal environment that is stable, a homeostatic environment, and in additionally, the removal of unwanted byproducts, unwanted foreign substances that will affect the total functioning of the animal. So all of these functions are carried out to maintain homeostasis, and the excretory system is an important player for the successful or for the what they call this, for the um, stable implementation of these key physiological activities. Okay, so let's emphasize on metabolic wastes. One of the major waste byproduct of animals across the kingdom, both invertebrates and vertebrates, are nitrogenous wastes, okay, which are attributable to proteins. So you would remember in our discussion during the midterm that proteins are made up of uh, building blocks, amino acids, and um, during metabolism or in metabolism, proteins are catabolized to these smaller building blocks, these amino acids. These amino acids are used to build protein internally inside the animal body. And in cases where there is excess concentration of these amino acids, um, the excess volume or concentration is not used by the animal in protein synthesis. They're not utilized, and rather they're broken down into simpler substances. They can be utilized as energy source. Some of them can be, or the breakdown components can be transformed or converted to other forms, to fats and carbohydrates. And there are different types of nitrogenous wastes that are filtered out and excreted by animals. And this includes the three listed here. So we have ammonia, urea, and uric acid. So we'll discuss each. But in general, in terms of the amount of energy needed to produce them, okay, uric acid is the most energy requiring. Producing uric acid is the most energy requiring, while ammonia is the least energy requiring. In terms of excretion and the dependence or requirement for water, for water to be present in order for these nitrogenous wastes to be excreted, ammonia is the one that will require more water for it to be excreted, while uric acid, since it's more soluble compared to the other two, will require less amount of water for it to be excreted by the animal. So, let's discuss each of these nitrogenous wastes. So, ammonia is a byproduct of um, many animals, including invertebrates such as protists and aquatic invertebrates, um, as well as vertebrates like fishes and amphibians. So ammonia, it's an inorganic compound, doesn't have carbon in it. So it's nitrogen and hydrogen. It's it's stable, and it's actually uh, more present, or yeah, it's present in gaseous form, okay, and has a very strong odor. It can irritate the soft tissues of an animal. So therefore, it has characteristic traits or features that deem it as a waste material that should not be allowed to concentrate in the animal body. Okay. Then we have urea, which is a combination of, or when ammonia is combined with carbon dioxide. Okay, so urea, found in the liver, it's water soluble, no, and it can be excreted via the urine. So the kidneys filter it out, and then it can be it can be concentrated in the urine and later on be excreted or expelled out of the animal body. And mostly in terms of the animals that produce urea, they're excreted also by vertebrates, mostly by vertebrates like, such as fishes, terrestrial amphibians, mammals, okay, kangaroos. We humans excrete urea, okay. Um, can also not Urea can also be found, aside from its presence in the blood. Urea can also be found in other bodily fluids such as the bile, our sweat. Urea can also be secreted or excreted via our sweat. Okay, and then we have uric acid. So uric acid primarily related to the guano excreted by birds and insects, reptiles, and land snails. It's a major nitrogenous waste that is um, derived from the breakdown of um, uh, what do you call this? A breakdown of purines. So the purine compounds such as those related the nitrogenous what do you call this? Um, the DNA building blocks, the purine DNA building blocks. So we relate uric acid to the breakdown of purines. No? So their uric acid is heterocyclic, so you can see in this illustration. So it's a cyclical compound and primarily attributed to the breakdown of purine nucleotides. So purine nucleotides, so your adenine and guanine, when they're broken down, um, the waste product, the waste byproduct is primarily uric acid. So uric acid can be um, the concentration of it in the animal body can increase if the animal is consuming uh, a diet that is rich in purine compounds. Okay? Right. So, those three nitrogenous wastes, okay, they are primarily present across, or they're present across the vertebrate and invertebrate groups of animals, and there are several structures across the animal groups that allow for the excretion of these nitrogenous wastes. So for the case of unicellular invertebrates such as protistans, they have a contractile vacuole. Okay, you can see here the one pointed, these two structures, spherical structures here, which are present in complex unicellular and multicellular freshwater organisms such as protozoa, can also be present in sponges, okay? And primarily the contractile vacuole controls water volume inside the animal, no? So the contractile vacuole collects and removes excess water that enters the animal constantly. So you can associate contractile vacuoles with animals that are of aquatic habitat. So for those animals, the contractile vacuole serves as the excretory organ. Then we also have in other multicellular invertebrates, okay, like the flatworms, we have um, the what do you call, or in general the uh, what do you call this, um, nephridial or nephron base excretory structures of these invertebrates. So we have the flame cell systems, the protonephridia, that's found primarily in flatworms, no? in related worms, such related multicellular invertebrates, such as tapeworms. So tapeworms and flatworms, they have flame cell systems. So these flame cell systems are simple and close type systems of Nepridia, which are branching. Okay, so the tubes run throughout the animal body, as you can see in the represent in the illustration here. So the tubules run across the body, and then eventually outside the organism, outside or on the opening towards the surface of the animal. So within the tubules, the flame cells are found, which contain cilia. So here you can see the blown up picture of a flame cell, and there's cilia associated to it. Okay, and the essentially water moves through these spaces, through these lumen. Okay, and filtering occurs. No, our filtering is primarily facilitated by these flame cells. Then for other related worms, invertebrate worms, so we have the metanephridia of an earthworm. So it's more advanced in comparison to the protonephridia of flatworms, and primarily composed of or has several structures. So there is the nepridiostome or the neprostome, okay, which is um, what do you call this? Which is a collecting funnel. So it serves as a collections part, a collecting part of the metanephridia. It collects fluid from the body cavity of the earth or the worm, no, the earthworm, and transports it to the coiled tube. So the nephridiostome opens to the body coelom and then collects fluid, allowing that fluid to enter the metanephridia proper. And then there's the coiled tubule, you can see here. Let's highlight. So the coiled tubule. Okay. Dismiss. So the coiled tubule here, you can see, right, which transports the fluid to the blood, to the bladder area. And then there is eventually the nepridiophore, which will secrete or will serve as the exit portal for waste material that has been filtered out of the fluid, the coelomic fluid of the worm. Okay? So essentially the direction of um, the direction of body fluid that's filtered, right, for the animal to, you know, remove the waste, to remove waste materials. So from the nepridiostome into the tubules, and then into the bladder area, okay, and then eventually out to the nepridiophore. Okay. All right, and then we also have the Malphigian tubules associated with insects, mostly our insects. So it's a set of um, tubules similar to the metanephridia, which is situated between the midgut and the hindgut of insects. So here you can see the structure, the yellow structure here highlighted. It's a Malphigian tubule, okay, and it collects waste material from the body fluid, from the hemolymph of the insects, and they excrete them together with the fecal material via the anus. So the waste material from the hemolymph is combined with the fecal material produced from the digestive tract of the insect, and that entire collection of waste, both from the digestive system and the excretory organ, the malphidian tubule, will be excreted together, okay, via the anus. Okay. And then we have for crustaceans, the antennal gland. So the antennal gland, it's present in crustaceans, also known as the green gland because of its color. And it's a paired gland found at the ventral part of the head of the animal, made up of several parts. So there is a sac for the filtration of blood. There is a coiled tubule for material reabsorption, for component reabsorption. There's a bladder that serves as a storage site that eventually empties through the excretory pore. So here you can see this blown up picture of the antennal gland found in crustaceans. Okay. All right. So now that we've covered the basic structure of excretory systems across different animal groups, mostly the invertebrates. Before we dive into discussing the excretory system of humans, no, let's review or have an introduction, an overview rather, of how excretion takes place. So down to the cell level, any waste byproduct will be transported out of cells into the interstitial fluid, the tissue fluid. Eventually, those waste material goes and diffuses to the blood circulating across different parts of the animal will definitely sequester more waste material in those parts, no? And key structures or parts of the animal body that dispose waste material or excretes waste material to the environment include, of course, for the digestive system, we have the gut. For the respiratory system, we have the lungs. Okay. Liver also indirectly or not completely, because mostly liver does a lot of metabolic processes, a lot of breakdown processes. Then there's the kidney for the production of urine, and the skin in some animals can serve as a site for waste excretion towards the external environment. And you've seen that, we've covered that. We have a number of examples in the invertebrate group of animals. Okay, so as a review, okay, the metabolic wastes are not only the nitrogenous based ones. There are also others that relate to too much temperature, too much heat. Okay, of course, for the case of the respiratory system, there's carbon dioxide produced during cellular respiration, which is not needed by the animal body. Excess water is also considered as a waste that need to be filtered out. And then bile salts and pigments from destruction of RBCs, the mineral salts that are not needed, and urea from protein metabolism are some of the basic, most common examples of metabolic wastes. No? Okay, so now we will look into the human urinary system. Okay, we'll look into the human excretory system. So we'll have an overview first of the structures. We're gonna emphasize more on the kidneys because it's a primary structure where excretion, or where the filtration of waste material happens, and concentration of those waste materials in the urine is facilitated. So what different structures comprise the human urinary system? So we have the kidneys, which is a paired set of organs, no? The ureter, which connects the kidneys to the bladder, and then finally we have the opening of the of the urinary system, which is the urethra. Okay, so we'll cover in a second in part two of this video. I will stop the recording to ensure that we have it's not going to be too long. So we'll cover in the next part the discussion on the human excretory system.